Great to see you all here tonight, and I think you're in for a really interesting talk. And I'm particularly looking forward to this because it's a subject that I need to know a lot more about than I know right now. So we have Luanika Mlera from the Rocky Mountain Labs. He's a postdoc fellow. His specialty is biology of vector-borne viruses. And he previously was a medical technologist in charge in Namibia at the Namibia Institute of Pathology. He has worked also as a medical laboratory scientist in public health laboratories of his home country, Zimbabwe. His education is at Northwest University in South Africa, where he has his PhD in biochemistry. An MSc in molecular biology from Staffordshire University in Trent, UK, and a diploma in bacteriology and a diploma in medical laboratory technology, both from the University of Zimbabwe. His research interest is in tick-borne flaviviruses and virus restriction, which I think we're going to hear more about tonight. So please join me in welcoming Luanika to this lecture series.
Well, thank you very much for the kind introduction, and also thank you for the opportunity to come out here and share some of my research work. I hope you find it as interesting as I do. I don't really have much of a choice because I work on it. I hope you will enjoy the talk. So today I'll talk to you about some work I've been doing to try and model what is called Powassan virus infection in what is called Paramiscus leucopus mice. So Powassan virus is a tick-borne flavivirus, and if you're familiar with viruses like Zika virus, which has caused a huge outbreak in Latin America recently, it's a cousin of those viruses. It's only the difference is that Powassan virus is transmitted by ticks, known as hard-bodied or exotic ticks. The Paramiscus leucopus mice, if that is strange, they are also commonly called the white-footed mouse, so maybe that's a little bit easier to make reference to. So I will start off by just going into a bit of basic biology about flaviviruses in general. Like I said, the Powassan virus is a tick-borne flavivirus. So these viruses generally have kind of the same structure. They have a genome that measures about 11 kilobases in length. And this is a positive sense RNA virus, which means that if you just take the viral RNA and put it in cells, the cells are going to translate that RNA into viral proteins, and you can actually rescue infectious virus just from the RNA. So the genome has a single open reading frame, which encodes for a single large polyprotein depicted in this schematic here. And this open reading frame encodes for 10 viral proteins. So the polyprotein is cleaved by viral as well as host proteases, or enzymes that cleave proteases into 3 structural proteins, known as C for the capsid, pre-M for the membrane, and E for the envelope. And we have 7 non-structural proteins. So the structural proteins make up the virus particle itself, as they are shown arranged in this schematic at the bottom here. The pre-M is cleaved, and the pre goes off like that, and then the M remains to have an infectious mature virus particle, as you can see on the right side. And when it is still pre-M, and it's an immature particle when it is on the particle like this, and it's assembled in that depiction there. So to infect cells, these viruses need to be able to get into the cells, and they are recognized by different receptors on the cell membrane. And in the case of the flaviviruses, they go into the cell but by what is known as clathrin-mediated endocytosis. and once inside the cell, the endosome is going to release the virus particle, which then loses the outer 3 structural proteins I've just shown you, to release the viral RNA, which is then transported to the endoplasmic reticulum, where translation of the message on the messenger RNA is going to be translated into the specific viral proteins that I've just shown you a moment ago. And it is on this endoplasmic reticulum that you find these small vesicles which contain various components of the virus, as well as some host cell material. And viral RNA synthesis occurs in these vesicles on the endoplasmic reticulum. And the pre-M cleavage that I showed you, where you have the pre being removed from the pre-M to end up with the M, actually is removed in the trans-Golgi network as you have a pH change, and then that leads to the formation of the mature particle. Then in an egress format, then the virus particle comes out, and then you can have the cycle continuing again. And in this endoplasmic reticulum, the messenger RNA or the viral RNA is weaved into the membrane and into the lamella, and as well in the same membranes as where you have this polyprotein being cleaved by the different viral as well as the host cleaving proteases into the individual protein structures I showed you. So like I said, the Powassan virus is a tick-borne flavivirus, and in the flavivirus family we have additional other viruses. I've mentioned Zika, which caused trouble in Latin America. But Powassan virus mainly circulates in the US. It's the only tick-borne flavivirus that circulates here in the US as well as in some parts of Canada. And it gets its name from a small town called Powassan, which is in Ontario, Canada, where they identified the virus in a 5-year-old boy who had fatal encephalitis. It actually killed him, and that's where the virus got its name from. But it also circulates in some parts of Russia, and in the same areas as well as Western Europe, you have what is called tick-borne encephalitis virus, and in general, all these viruses would cause encephalitis, which is inflammation of the brain. Yellow fever is also currently causing trouble in Latin America. It's killing off some monkeys, and it has spilled over into humans as well. And I think there's probably more than 1,000 cases of human infections now. And dengue virus also is transmitted by mosquitoes. So I mentioned that Powassan virus is the only tick-borne flavivirus that circulates in Northern America. So in the US, you find that this virus circulates in these areas that are depicted in the yellow color here. That's where most of the human cases occur in the US, although there are not as many cases as we find with other flaviviruses like your mosquito-borne, but it does cause very severe infection in humans, and it can also lead to death. So in nature, these viruses are maintained in a persistent cycle that involves infected, exoded, or hard-bodied ticks, like I mentioned. But what is interesting is that once infected, these ticks are infected for the duration of their lives, and this can span from 2 to 3 years. And they can transmit the virus to their progeny through a transovarial transmission, and through the different stages of development of the tick, they maintain the virus, they never lose it. And what's also amazing is that when they obtain a blood meal from small to medium-sized mammals, such as rodents and skunks and the deer and so forth, when they feed in close proximity on the same host, they are able to transmit the virus from one tick to another, and we call this process co-feeding. And these small mammals play an important role in the ecology of the virus, in that as the ticks are feeding on them, they are also releasing the virus into a different mammalian system, which is different from the tick that normally carries the virus. And so these mammals may have various roles. One could be that they can become reservoirs of the virus. They can just be bridge hosts in which the virus is there transiently or for a short time before it is carried by a transmitting vector to another host. But that I should emphasize is the importance of these small mammals. This is where we are coming from, trying to understand what is the relationship between Powassan virus and these small mammals that we find in nature. So humans get infected mainly through tick bites when they go out into the woods, and then they come across infected ticks, and then that's the main route of transmission. But in other places where tick-borne encephalitis viruses are also endemic, places like Hungary and other parts of Europe, it is also possible to get alimentary infection through the consumption of unpasteurized milk from domesticated animals that may be shedding the virus into the milk. So it's a dynamic process there. And once infected, the incubation in humans varies from 1 to 5 weeks, and before you can be able to see the symptoms of infection. And the symptoms include fever, a headache, vomiting in some cases, as well as weakness. And once the virus is found in the brain, then you have encephalitis, which is inflammation of the brain itself. You can also get meningitis, which is inflammation of the membranes that surround the brain, or the meninges. And as a result, you end up with confusion. You can get loss of coordination because your motor facilities or faculties are being affected. And in some cases, you can have speech difficulties and seizures. One aspect of these viruses that I'm also interested in, and I've published a little bit about it, is that at least 50% of the survivors of the encephalitic phase end up with chronic neurological infection or neurological problems. And that could be persistence of the virus in the human after the primary infection. So maybe in some cases, some people may not clear the virus completely, but in some cases, it may be because of damage that occurred to the central nervous system during the acute phase of the infection. And then you have these long-term neurological symptoms. So specifically for Powassan virus, the case fatality ratio is about 10 to 15%. And like for many other flaviviruses that cause problems in humans, there are no licensed drugs to be able to treat these infections. I'm going to be talking about development of the Peromyscus leucopus mouse as a model for studying Powassan virus infection. That is important because the Peromyscus leucopus are a natural host, so they are different from the BAUCs that we have as an inbred laboratory mouse that we can use to study in the lab. So these have been bred in the lab over periods of time, and they may have some genetic differences from what is currently in the wild. But the BAUC mouse model is an important model because it recapitulates the kind of symptoms that you see in humans when they get infected. Once infected, usually they die by 8 days post-infection, and they also present the same neurological signs such as hind limb paralysis. They lose their weight, and eventually they are killed. One group has reported that respiratory insufficiency contributes towards the death mechanism, so the animals have difficulty to breathe, and the amount of air they take in is not sufficient for them to continue living with an infection. Another group at UTMB in Texas also showed that when they use ticks to feed on the mice, then the mice are going to mount an inflammatory cytokine response at the skin interface. But in this model, that is not very useful because the animals still go and die. Because the viruses are transmitted by ticks, when the ticks are biting, they are also spitting out their tick saliva, and this tick saliva has various components that also modulate your immune response, and therefore the tick saliva is important in enhancing transmission and also influences the cause of disease. So in cases where you use tick saliva to infect, you get very much more severe signs of disease after that. So the Peromyscus are a wild species, like I said, and we do have evidence of tick-borne flavivirus infection in various types of mammals and small mammals in nature. And here I'm showing you, for example, for the Peromyscus species, we have been able to detect antibodies that are specifically targeting this virus, which means that these mice have been naturally exposed and then they developed an immune response to the virus. However, we have not yet been able to isolate infectious virus from the Peromyscus species. In the voles or the Myodis species, we have been able to show the presence of antibodies against Powassan virus, but there's also been additional evidence of antibodies specifically against the tick-borne encephalitis virus, although virus has not also been isolated in this case. And in the eastern part of Asia and some parts of Europe, what is called the yellow-necked mouse are depicted in this last column here, also known as the Apodemos species. In this case, they've been able to show the presence of antibodies and also to be able to isolate infectious virus from the spleen and the brains of these wild-caught mice. So that is very substantial evidence that in this case of the Apodemos mice, they can indeed act as reservoirs of the tick-borne encephalitis viruses. And so based on this, we don't really have much of an understanding of how these mice respond when they are exposed to the virus. We can only tell that they have had antibodies, which means they've been exposed at some point. But how do they really respond and how do they cope? So that's one of the questions I was trying to ask with some of my studies. Before I show you some of the results, I just wanted to bring to your attention why we think the Peromyscus could be important for the US context. The Peromyscus are the most abundant mixed forest rodent in the US, especially the eastern part of the country, where in gray, depicted in gray here, you have the Peromyscus maniculatus, and that spills over into Canada as well. And you also have the Peromyscus leucopus, which is the species I'm working with. And you can tell from here that the distribution of these small mammals also coincides with the distribution of the human cases that we have been able to record for the US. So maybe these mice could be playing a role in nature, we don't know until we do the studies. And they may not be the only mice that are responsible, there are additional other small mammals on which the ticks feed on. And on the right side here, I'm just showing you how different the Peromyscus genetics is from the moose or the Bauxi mice that we use in the lab. So they are fairly divergent. They are much more closely related to the meadow vole as well as the hamsters. So now I'm going to talk about how I established the... Is it still on? Can you hear me now? Yes. Okay. I felt like I was shouting a lot. Okay, so now I will talk about how I established this Peromyscus leucopus mouse model. But it was also based on this publication in which they had collected ticks from deer and then isolated the virus and used that to infect neonatal brains of mice, and then after amplifying the virus, then they use that to infect additional mice as well in the lab. So after their experiments, they found that after infecting the white-footed mouse or the Peromyscus leucopus, there was no sign of infection at all, no sign of clinical disease, and they thought that maybe this ability to survive the infection could suggest that the white-footed mice or the Peromyscus leucopus could serve as reservoirs for the virus in nature. So we wanted additional information to be able to substantiate this. So what I did was to infect 3 mouse species, the Peromyscus leucopus, the Bauxis, and the black sex mice. Then these 2 are what we normally use in the lab. So I infected with 1,000 particle-forming units of the virus, and this was by intraperitoneal injection, which is just above the leg, below the abdomen there. And we set the experimental endpoint at 28 days to see what happens to the mice when they're infected. So what I found was that the Peromyscus leucopus that were infected via the intraperitoneal route, they survived the experimental endpoint as shown by this green line, and they survived without any signs of disease at all. But in contrast, the Bauxi mice, they also succumbed by day 8 as expected because of very severe neurological disease. And interestingly, the black sex mice were fairly partially resistant to the infection. Some of them died and some of them survived to the experimental endpoint. And for the Peromyscus leucopus mice, they never lost any weight, but for the Bauxis, you can begin to see some weight losses in infected mice from about day 6. And for the black sex mice, you can see loss of weight in these mice, and the ones that recovered and survived to the experimental endpoint are shown going up again there. And what was interesting was that I could find viral RNA after an intraperitoneal infection. The virus is neurotropic, which means it prefers to infect tissue of central nervous system. In the brain, we can now be able to detect viral RNA in high amounts in the biopsies and in the black 6 mice, but we're not able to detect any viral RNA in the brains of the Peromyscus leucopus mice. So we thought that maybe the Peromyscus leucopus are peripherally resistant to the infection because we're putting it IP. And we know that the virus needs or prefers to infect tissue from the brain. So what we did next was to challenge the mice again, but put the virus directly into their brains and hope to see disease. And we set the experimental endpoint again at 28 days. And what was surprising for this very drastic challenge in the Peromyscus leucopus mice is that they also survived the experimental endpoint of 28 days, and they did that without any clinical signs of disease. So they were pretty tough mice. But for the biopsies and the black 6, intracranial inoculation was very lethal for them, and they died at either day 4 or day 5 after infection. So there's this huge difference between these mouse species. And what I found was huge amounts of viral RNA in the brains of the mice that died, the biopsies and the black 6s. And we're able to detect now some smaller amounts of viral RNA in the brains of the Peromyscus leucopus mice, but this was at day 28, which suggested that even though there could have been replication of the virus in the brain, it didn't really cause much effect on the mice. And so we wanted to look at this more closely. And we see that distribution of these viral RNA, so these are brain sections from the biopsies and the black 6 mice. You see that the virus is all over the brain as well as in the spinal cords of these mice that are going to die. But at day 28 in the Peromyscus leucopus mice, we are not able to detect the viral RNA. And the viral RNA is depicted as a brown color in this image here. We also looked for pathological signs in the mice, and we found that in the mice that die, the black 6 as well as a biopsy, they have what is called perivascular cuffing, which is an infiltration of lymphocytes around blood vessels. So that's part of the encephalitis or inflammation you get in the brain. And interestingly, we're also seeing some but very mild inflammation in the Peromyscus leucopus mice, but this will be at very early time points. However, when we look closely in the brains and the meninges, we see that there's meningitis in the black 6 as well as the biopsy mice, but the meningitis is absent in the Peromyscus leucopus mice. But it was puzzling and interesting that we do get some mild inflammatory response in the central nervous system of the Peromyscus leucopus mice when challenged with Powassan virus at early time points. I was also curious to know if the Peromyscus leucopus were actually actively responding to the infection and whether they were whether they were seroconverting to the challenge. And in this panel, I'm just showing you what the virus does to cells that are infected in the presence of mouse serum from controls that were not infected with the virus. So they don't have antibodies, so they cannot restrict the virus infection here. But the serum from infected mice is able to restrict the virus growth when diluted 1 in 10 or 1 in 50, and we begin to see a little bit of virus coming up at a 1 in 250 dilution. So this tells us that the antibodies that are being generated by these mice are actually neutralizing, so they are able to bind the virus and prevent it from infecting. And the more you make them more dilute, the more they become less effective. And we concluded from this assay that the focus reduction, the foci being these little plaques that you see, required to inhibit 60% of the virus was a dilution of 250. So indeed, the Peromyscus leucopus mice do seroconvert and generate antibodies against the Powassan virus. I indicated earlier on that we get very small amounts of viral RNA at day 28 after infection, but we wanted to know what happens at earlier time points. So I infected the Peromyscus leucopus, challenged them again intracranially or into their brains, and then euthanized the mice from day 1, day 3, 5, 7, and 15, and then did some viral assays to see how much virus we get out of their brains. And so this graph shows you on the left how much virus is coming out of the homogeneous from the brain, and this is infectious virus, and this is a depiction of how much viral RNA we can detect in the brain as well. So the fact that we see infectious virus coming out means that the virus is not being totally cleared in the early time points. But what you should appreciate from this graph is that there's no dramatic increase either in the amount of virus that we are seeing. It kind of remains more or less the same. It doesn't really go to a peak like we see with other viruses coming into the brain. What was also interesting was we could detect viral RNA in the brain, in the blood, after having inoculated the virus into the brain, which meant that the virus was able to leak out into the blood system. But this was mainly between day 1 and day 4 after infection. And therefore, this could mean that this would be the best time for any tick to come and feed on a mouse infected in this manner and be able to get infection and then be able to transmit the virus. But at later time points, it seems like the virus gets cleared from the blood. So we were curious to know where was this viral RNA in the brains of the Peromyscus leucopus mice. We could detect by qPCR that we're getting around 1,000 copies of the genome. And I showed you earlier images where we had viral RNA all over the brains of the Peromyscus, of the biopsies and the black 6 mice. But interestingly, in the Peromyscus leucopus mice, after a challenge with intracranial inoculation, we find that the viral RNA is mainly restricted to the olfactory bulb here. The olfactory bulb is responsible for your sensing of smell. So it seems the virus is restricted just to the olfactory bulb and tracks back into the ventricle like this. This is just a zoom in of that lip area. We do see smaller amounts though in other parts of the brain, like the cerebellum and the cortex, but not as much as we see in the olfactory bulb. And the panel below shows you, I mentioned that the viral RNA is a positive sense, but when it's replicating, it's the only time that you can begin to pick what is called the negative sense of the RNA. So it's a template on which you're going to make the positive sense RNA. And so that's an indication for us that the virus is actually replicating. So we needed to know that the presence of this RNA in this place of the brain is the virus actually replicating. And indeed, we can show that it is replicating, but still restricted to the olfactory bulb. And in contrast, for the biopsies and the black 6, replication is all over the brain, as indicated in this image. We also do get some viral RNA in the spinal cord as well. So to summarize this part, I told you that we do not see any overt clinical signs of disease in the Peromyscus leucopus mice when you inject them either IP or intracranially with Powassan virus. We do, however, see mild encephalitis after intracranial inoculation, and Powassan virus replication is restricted to the olfactory bulb and the ventricle. The Peromyscus leucopus mice, they seroconvert, meaning that they do generate antibodies targeting specifically the virus that we have put into them. However, the biopsy mice succumb to intracranial and IP inoculation at early time points. The biopsy mice seem to be partially resistant to intraperitoneal inoculation, and this could suggest possibly persistence of the virus in these mice, and we would need to do additional studies to actually show that. Now I've shown you where some of those experiments took us to, where we found that there's no disease really in the Peromyscus leucopus mice, but we do find some mild inflammation, and we wanted to be able to characterize this specific host response, and this is what I'm going to talk about next in this part of my talk. So the experimental design was more or less the same. So we inoculated the Peromyscus leucopus with 1,000 particle-forming units of Powassan virus, and we put the virus directly into the brain. And then we euthanized the animals at the time points shown here. And what I did next was to do a brain transcriptome deep sequencing so that we can be able to see the genes that are involved or that are provoked by the virus. So what was interesting from this study was that indeed we find some genes being significantly differentially expressed, either being upregulated or being suppressed in the expression. And depending on how you set your parameters for this analysis, we found that at 1 day post-infection, if you decided to just look at genes that go up by either twofold or go down by twofold, we would have 42 genes at 1 day post-infection. And the number increases over time, and I think this graph shows that increase nicely, that by day 7 post-infection, you have the maximum number of genes that you can get that are significantly differentially upregulated or downregulated as a response to the virus. And then there's a downward trend in the number of genes that you're finding in later time points in the infection. So this suggests that there is sort of a deliberate and calculated way to respond specifically to this virus. So I needed to analyze what were the genes and what could they be doing in terms of responding to the virus. So I used what is called ingenuity pathway, which is basically a software in which you load your genes that are either going up or going down. And then these genes are processed into and classified into different pathways of responses that we can be able to put them into. And some of the pathways that were coming up as important pathways involve interferon signaling, and you might know that interferon is an important factor that is produced in response to infection. And we also had activation of interferon regulatory factors by cytosolic pathogen recognition receptors. So these receptors, they sense viral RNA and then they trigger a cascade of responses that at the end you result with the generation or the secretion of the interferon and a lot more other signaling pathways involved. And for some of these pathways that are indicated in red, we would pick that pathway for at least 2 times, but different time points after the infection. For example, interferon signaling would detect that pathway at day 1 post-infection, as well as 3 days post-infection. So if you want to be fancy, you can then be able to plot the kind of network or pathway that you are actually looking at and see, in this case, this is just interferon signaling, and we are looking at these genes in orange. They are sort of the genes responsible for the upregulation or the overall picture we are seeing. So most of these are interferon regulatory factors, and in the periphery of the network in red are the interferon stimulated genes that are upregulated in response to the infection. And what you see in green is a gene that is actually downregulated, and in blue is a pathway that is being inhibited in this case. And to show the same data differently, we would look for what are called RIG-like receptors, like your RNA recognition molecules, and how they are changing over time. And we see that there's this trend that you have an upward increase in the expression of the genes over time for a molecule called RIG-I, for example, or LGP2 and MDA5, and all these do sense foreign RNA when it comes into the cells and then trigger the cascade to produce the interferons. And another pathway is what is called the toll-like receptors, which also sense viral RNA in vesicles, and then you have similar patterns of gene expression. And this is also for interferon regulatory factors, which are responsible for the control of how much interferon you are going to have in the cells. Too much will be detrimental, and too little might not be as effective. If you look closely at what are called ISGs, or interferon-stimulated genes, which are genes that are upregulated in response to the secretion of interferon, we see here, for example, something called IFIT2 or IFIT3, and they all go up over time. And the trims are what are called tripartite containing motifs, and they're also known as restriction factors for some of the viruses like HIV, for example. And these have been shown to be effective against viruses like HIV. This thing is going off on me every time. So we did most of our studies in vivo in the mouse, but we are also able to get peromyscus cells that we can also be able to work with in vitro in a better controlled situation. I should say the peromyscus are pretty feisty to work with, so you have to sedate them. And when you work with them, they are pretty feisty, and it's easier to work with cells in a flask because nothing is going to try to bite you. But what's interesting here in this graph is that I have cells derived from the black 6 mice which are going to die following infection, and I also have cells derived from the peromyscus mice which we know are resistant to the infection. And the difference between the two is that in the mice that are going to die, there is increase in virus replication if you infect these cells in vitro in culture. But the cells derived from the Peromyscus seems to be refractile. They don't really produce much virus. It actually goes down to undetectable levels. And this kind of mirrors what we see when we infect the mice, that we don't really get clinical signs of disease. We don't really get huge amounts of virus coming out of these mice. So this could be a nice model to actually look at those genes that I was showing you and say, can we knock out some of these genes in these cells rather than in the mouse, and be able to do some studies with that. And this is also kind of a similar depiction, but we use what is called confocal microscopy to look for viral antigens. In this case, the envelope protein and the NS1, non-structural protein 1, NS3, and NS5. These two are depicted in red. So where you have the red signal, I don't know if you can see it very well at the back there, but where you have the red signal, it shows you that's the NS1 being expressed in the cells. And in white here is the NS3, non-structural protein 3, and NS5, which is a viral protein that is also involved in antagonizing the interferon response. So in the black 6 mice, in the fibroblasts that we have on them, we do get nice evidence that the virus grows very well in those cells. But if we contrast this with the cells that we get from the Peromyscus leucopus mice, you see that there's basically no red signal or white signal, in addition supporting the fact that we don't really get virus replication in the Peromyscus leucopus mice cells. So I told you that we basically look at a number of genes over different time points, and then you can look very closely at these genes and try to make sense of what is happening across those genes. So when I looked at day 1, some of the genes that were very much upregulated included something called KRT20, which is a cytokeratin encoding gene. We also had something called SP100-like, and this is an interesting gene that I found in these studies. And so it is upregulated, indicated by the red color there, and the green shows that the expression of the gene is suppressed. So I was curious to know what this gene was doing in the context of this infection. So using those cells now in culture, we could knock out that gene. And if you placed it in the map, you would see that the SP100 comes up as a very highly upregulated gene, and it forms part of this interferon signaling pathway, which suggests that the appearance of this gene in particular is responsive to interferon signaling or interferon induction. So that SP100 protein belongs to a family of proteins that are called PML nuclear bodies. They are many different types, and that one particularly is called SP100 because it is speckled. When you stain the cells, it comes up as speckles, and it weighs about 100 kilodaltons. That's where it gets its name from. And it was, some of these were associated with alpha thalassemia as well as promyelocytic leukemia, which is where the PML comes from. And these proteins are induced by interferon in some cases, but in some cases they are constitutively expressed, so they become what is called the intrinsic immunity. It is just there and it will respond or bind to viral and foreign antigens. And they have very diverse functions, which include repression of transcription. They bind certain motifs on DNA and affect expression of genes. And in some cases, they are also antiviral. So in the case of DNA viruses, we have a number of viruses that are known, for example, to be associated with these PML bodies. For example, adenoviruses, HSV1 or HCMV, and these, because they have a replication cycle that is nuclear because they are DNA viruses, then they can be targeted by these proteins. And what these proteins do, in some cases, is actually to surround the virus particle and prevent replication of that virus. In some cases, some viral proteins are degraded, so it's not available for continual viral expression. And in the case of RNA viruses, we have, for example, dengue virus. The C protein, the capsid protein, goes in and out of the nucleus, and it can be targeted by what is called DAX, which is also part of the PML nuclear body, in which we find the SP100 going up as well. additional evidence has been shown for flaviviruses that if you knocked out PML, for example, in this case, you actually increase viral replication, for example, using siRNAs or small interfering RNAs targeting the gene that you are looking at, then you have increase in the virus. But when you overexpress the protein of interest, which is having a restriction factor on the virus, then you don't really have virus replication going on there. So in my case, what I did was to knock out a number of genes, including the SP100. And I showed you the graph where in Peromyscus leucopus mice, if you just infected the cells, you don't really get virus coming out of them that much. But in this case, when you knock out the SP100 or knock down the SP100 gene using small interfering RNAs, we found that there was now an increase in the amount of virus that was coming from the very same cells. But when we knock out a few other genes like KRT20, which was also upregulated in that case, or SP110 or PML, it really didn't have much of an effect on the virus replication. This suggests that SP100 might be playing a very potent role in restricting replication of Powassan virus in these mice. So it is a very interesting result that we found, and I'm busy working on trying to figure it out and determine the mechanism by which this SP100 could be restricting virus infection. So to summarize this part, I have told you that the Peromyscus leucopus mice mount a robust and well-controlled interferon response. And it seems like the SP100 gene is upregulated in response to Powassan virus infection. In addition, if you knock down that gene, then Powassan virus replication is going to increase in the cells. However, the knockdown of PML, KRT20 or SP110 had no effect on Powassan virus infection, suggesting that maybe it's specific to just the SP100 gene that I've showed you. So I'm currently now working to expand the work on the SP100 protein or gene and see what we can do. We can knock it out and then be able to knock it out completely. The siRNAs just decrease or suppress the amount of expression, but CRISPR technology can be able to knock it out completely. Then we expect an increase in the amount of virus from what I showed you earlier on. And we will also try to overexpress the SP100 and in particular in the cells that we know are coming from mice that are going to die. Can we be able to prevent cell death in those cells when we overexpress the SP100, then confirming that it indeed is having a restriction factor role in the cells. We would also be interested to find whether the SP100 would have an effect on other flaviviruses. Ask the question, would it affect other flaviviruses like the mosquito-borne flaviviruses, for example, and could that be applied in the long run as a therapeutic agent, or can we find small molecules that we can be able to use to stimulate the expression of SP100 in the case of infection and use that as a therapeutic. And I'm also looking at trying to understand a bit more of the differences in interferon signaling that we find between the black 6 mice that go to die. We think that they do respond, but maybe the response is not the same as what we see in the Peromyscus leucopus mice, so we need to tease that out. So in conclusion, I would like to just thank members of the biology of vector-borne viruses section, in which I belong in the laboratory of virology at the Rocky Mountain Labs, particularly Danielle Offerdahl, who did some of the work that I showed you, particularly the confocal images where we detect viral proteins. And I would also like to thank Marshall Bloom, who is the chief of this section, for providing me this opportunity to do the research in his lab. I would also like to thank people in the genomics unit who are very helpful in doing the next generation sequencing and transcriptome data analysis that I've showed you too. And people from the Rocky Mountain Veterinary Branch, particularly Kimberly Mead White, who helps out with infections and necropsies of the mice in the lab. I would also want to thank Heinz Feldman, being the chief of the lab of virology, and Joe Prescott, who has used a different version of the mice in studying hantaviruses. And with that, I'll be happy to take any questions you might have. Thank you.
Let me ask you first, this is a very systematic and detailed study. How long did it take you to get to this point, and how long do you think it's going to take you for the work that you want to do in the future?
I would say it took probably about 3 years to get to where we are now. When we started off, I had a bet with my boss that he said the mice are all going to die, and I said, well, they won't all die. I had my own percentage, but we were both wrong. The challenge with the mice, we do have a colony at the lab, so we can breed them, but the challenge is that they are very divergent from the other species of mice that we can work with. So the tools to be able to study different aspects in the Peromyscus are not readily available. For example, antibodies against most of the proteins we can find in the mice, but we cannot really find for the Peromyscus leucopus. So one example is when I was looking at whether the mice were actually seroconverting or not. When we did ELISAs, where you use a secondary antibody to bind the primary antibody coming from the mouse. So we use an anti-mouse antibody on the market, and it didn't work. And when you looked at the results, it looked like the mice were actually not seroconverting until we used a very specific secondary antibody against it was the anti-Peromyscus. Then we were able to now show that they do indeed seroconvert. So that took like maybe 6 months or so to achieve. And another challenge with the Peromyscus is that they breed slowly, especially if it's winter. For some reason, no matter how we try to change the environment to feel like it's a natural environment, but I think for some reason they have a biological clock to know that it is winter, and then they just don't breed as much. And then you have to wait to get the adequate numbers to be able to do the experiment, so that took a bit of a while. But I'm happy to say that for some of the part I've just published that, so we are now working on the genetics now, and maybe in the next 2 years we could have hopefully some answer. So we have antibodies that we have, like for the SP100, for example, if you just use antibodies for the mouse, they will not work. So we have to have these custom made, and it takes several months for those just to be made, and it's also fairly expensive.
So is there down the road, is there a possibility that you could come up with some kind of a vaccination that would be useful for humans?
There's actually some licensed vaccines for the tick-borne flaviviruses, but it's not licensed to the US market yet. This is available in Austria and some of those countries in Europe. In the US, we are currently undergoing some clinical trials for the same vaccine. There was some vaccine which had been developed, I think, in the mid-80s or 90s, using one of the attenuated strains of the tick-borne flaviviruses. But I think there was a case, an adverse event in, I think, about 1 in 18,000 cases, and for that reason it was discontinued. But in some countries like Russia, the Czech Republic, they do have a vaccine readily available, especially for the lab workers, so they can get vaccinated. We are still a little bit behind. So our work, I think, is probably not looking at a vaccine for these, because it's there and it's just maybe licensing it in different countries. But maybe what could be important is to develop some drugs that could be used, maybe, because if you get an infection, it might be too late to actually get a vaccine, so you just need something that's going to knock out the virus as soon as possible. So maybe that area will be more interesting for me to pursue.
I'm curious about the CRISPR knockout process. Is that, like, how effective is that in fighting diseases in animal population? Like, does it take a long time? Is it expensive? Like, how is
You
that?
mean to use it in the wild? Well, there's different applications you could do with the CRISPR technology. It is very effective in terms of knocking out the genes. You may occasionally get some what they call off-target effects, where because what you have basically is what is called a guide RNA that is going to guide the enzyme called the Cas9 to the specific site where you want it to cut out. So the enzyme is going to cut a part of the gene, and then your cell is going to try and repair that excision. And in doing that, it does what is called a non-homologous end joining. And then, so that messes up the gene. So for gene therapy, there are people who have trying to use it like for HIV therapy, for example, I think it's still a little bit of a long way from clinical application to a certain extent. You need a lot of validations and specificity. You don't want those off-targets happening in the human, for example. In the wild, in animals, it's probably not what you would want to do in wild animals, but we use it in the lab because we just want to knock out genes and see a phenotype or no phenotype. And then we have a colleague who is trying to use some technology to sort of bait the ticks, for example, and then bring them to where they, take up something that's going to actually mess them up and then not be able to take up or clear the virus infection from them. Maybe something like that could be applicable, like in a small area where you know that you have a lot of ticks, and you could apply it in that way and then just prevent transmission into humans.
Question?
How do you prevent the infection to you? You said the mice get bit by one of the mice that are infected.
Well, I should have mentioned we work under what is called animal biosafety level 3. So you have additional levels of protection for Powassan virus. So there are procedures and protocols if accidents happen of what you should do. One of them, for example, if it bit you on your finger, you have to express the blood for 15 minutes under running water with scrubbing all the time. We know there's basically no treatment, but you will be monitored for what could happen next. But we try as much as possible. When you actually do the inoculations with needles and stuff like that, the animals are actually sedated, so they are knocked out. And then you do all your procedures, and then they wake up, and then you put them in the cages and you put them away. And for most of the procedures that are sort of invasive or where you use needles to get into the mice, you need to have them sedated like that. They are also screened, like when they are not infected, they are with the virus specifically, they are also screened for some pathogens that could infect mice. So I think that is done quarterly, and they are always negative for that. So we hope that the main thing is just to avoid being bitten. But before you sedate them, you have to pick them up. So we have forceps, and the length of the forceps is going to be useful in when the animal tries to clasp back at you. If the forceps is this long, then it really doesn't have room to get to your hand. But if it is a short forceps like this and you're holding it like that, then it's pretty close, and it could. So it's things like that that you consider when you're working with the mice. So far, we haven't had incidents of being bitten.
So the virus is concentrated in the olfactory bulb. Was there any effect on the smell of the mice?
That's one very good question, and we wanted to ask the same thing, but when the mice have impairment of their olfactory sensation, they are likely to not move towards food, for example, because they can't really smell it. But in this case, they seem to be okay after all this infection. They eat and their weights go up. So, but it could also be because they are contained in small cages. Before you actually infect them, we acclimatize them to that cage. So maybe they get used to where the food is, and they could easily find it. So we really can't test, or we really can't tell just from a behavioral point of view that their sensation has been affected. But you could do specific tests. For example, put in, you remove them from the normal cage, and then you put them in a different cage where there's some scent that they would like to have, like maybe peanut butter flavored scent. And if they go towards it, then you know they can smell it, or maybe they don't go toward it and they can't smell it. And you can also use certain chemicals that should be repulsive to them, and that when you put them in the cage, do they hang around a repulsive smell and then are not able to sense the smell, or they move away from it. So it's experiments like that that you could do, but we really haven't done the experiments to determine if there's a specific impairment of the olfactory sensation. But it's a good question.
I'm just just wondering, do they like, I don't know if you've studied this, but do they target the olfactory cells specifically because they're more susceptible or more permissive? Like,
That's also a very good question. We do not know at the moment why the virus ends up in the olfactory bulb, but it has been shown for some other flaviviruses that even if infected subcutaneous, for example, away from the central nervous system, the first part of the brain that you're going to see virus for these neurotropic viruses is the olfactory bulb. So we don't, and some people think that maybe it's just the circulation that carries it to that area. But in this case, it could be that the cells in the olfactory bulb are probably much more susceptible to the infection than the other parts. Maybe the expression of the SP100 is not the same in the olfactory bulb versus the other parts of the brain too. We don't know.
Well, this has been a very interesting talk. I think you've learned a lot about the work that he's done, but also about the very systematic procedure that is involved in this kind of scientific research. And I think we're really fortunate that we are so close to the Rocky Mountain Lab, and we can have specialists like Luke and Leela come and talk to us about his work. It's really cutting-edge kinds of research that he's doing, and so I want to thank him again for taking time to come up here. And if you have any further questions that you'd like to ask him on an individual basis, you're welcome to do so. So thank you very much. Next week, we're going to have a talk. It's going to be kind of interesting. We have two people who are going to talk on giver medical care mission in Tanzania. And they are also bringing with them the bishop that they work with in Tanzania. And he just happens to be here at Missoula. So we're going to have the three of them talk to us about that experience next week. So hopefully see you all here then again.